Number 6 says, complete the punnets for the following. A female X-linked recessive carrier crossed with a normal male. Okay, so do this one on the board. Female X-linked recessive. We got, I'll just put T's on here for the trait. So recessive will have 2 lowercase t's on the X-linked trait. So the female's going to have 2 of these. Just like this. Okay, and a normal male. So normal male is just going to be XY. Okay. Okay. So you've got 2 males. Nope, you've got, oh, I messed that one up. This is a Y. Okay, so you've got 2 males that express the trait, and then you have 2 female carriers. Okay, so the males they express the trait. They only have one X. Okay, so they're XY. So if they have it on that X, they are expressing that trait. They are expressing that disorder or whatever sex-linked disorder it is. And then since females have 2 X's, they have to have it is recessive, so they both have to have that to express it. So these 2 are carriers, okay? Because only one X has that trait. Okay. The next one says X-linked colorblind female and a normal male. So if we just change the trait, so X-linked colorblind female and a normal male. Okay, so we have the same results here. You have 2 females that are carriers, and you have 2 colorblind males. Okay. All right, number 7. Well, it says what phenotype patterns are seen in different kinds of crosses. Okay, so in different kinds of crosses. So in monohybrid crosses, you like in the sex-linked ones, you have a 50-50 chance. Well, if you're female, you have a 100% chance of being a carrier of colorblindness, and if you're male, you have a 100% chance of being colorblind. Not really sure what exactly it's referring to on that question, but we'll go on to number 7. Okay, homozygous dominant and homozygous recessive. This is a monohybrid cross, and it wants to know the genotypes and phenotypes for these. So you have homozygous dominant. We'll just use ours. Remember, homozygous dominant is you have 2 of the same alleles, and they are both dominant, so they're both uppercase. And then we are crossing that with a heterozygous, which is going to have 2 different alleles. a dominant and a recessive. Alright, so I set up the cross. Here's my heterozygous, here's my homozygous dominant. So we have one that is going to also be homozygous dominant, two homozygous dominant, one heterozygous, two heterozygous. So genotypes here are homozygous dominant and heterozygous. Okay, the phenotype for all of these are going to express the dominant allele. Okay, so they're all going to be dominant phenotype. Okay. Even though this is the genotype, it is heterozygous and it does have this recessive trait. Recessive means it does not show up unless you have two recessive alleles. Okay. But if you have a heterozygous alleles, then the dominant one is the one that gets expressed in the phenotype. Okay. Number 9, homozygous dominant and homozygous recessive. This is dihybrid. So if we are doing a dihybrid cross between homozygous dominant and homozygous recessive, we'll do big R, big T times little, or times cross with little r, little t. Okay, so remember there's 16 squares to this. So the way we do this is the full method. Okay, so to get our different combinations for just this one, we do the first in the genotype, so that's RT. You do outside, which is also RT. Okay, and you do inside, which would also be RT, capital RT. And then last, which is going to be two last ones. So they're all going to be capital R, capital T. Let me rewrite that. Okay. And it's going to be the same way for this one. Okay, so all of these different combinations is going to be lowercase r, lowercase t. Okay. Now, again, all of these combinations are going to be the same, which ultimately is going to be heterozygous. Okay, so you have big R, little r, big T, little t. It's going to be the exact same thing for each one. Okay, so all 100% of the offspring for this cross will result in that genotype, and they will all have the same phenotype. Okay, so for number 9, genotype is going to be, we'll say big R, big T, little t. And the phenotype is going to be dominant because you have a dominant allele that's, it masks this recessive gene. You also have a dominant allele here that masks this one. Okay, so they are going to be dominant phenotypes. Okay. Number 10. If you cross a heterozygous and a heterozygous, this one's a little bit more difficult. Okay. Heterozygous and heterozygous. Now we'll stick with the same letters here. I'm going to move my board this way. So we have this is what we're crossing. We've got heterozygous and heterozygous. Okay. And we'll get the different combinations. And once we get the combinations for one, we'll have the combination for the other. So we only have to do this once. So first, we have capital RT. Outside, we have capital R little t. Inside, we have little r big T. And then last, we have little r little t. And that's what it's going to be for the other one. So we'll go ahead and write those on the side. RT. Whoops. Okay. Now, this is what it should look like. I can zoom out that. There you go. If you need to pause anytime during this video, just pause it. Different combinations here. And what it's want to know is the number of times that this phenotype shows up. And you have 4 different phenotypes from this cross. Okay. So you have homozygous dominant. Okay, there are 9 that will show the phenotype of both traits as dominant. Okay, you have 3 out of 16 that will show up as dominant for the first trait and recessive as for the second trait. You have 3 that is going to show recessive for the first trait and dominant for the second trait. And then you have 1 that is going to show both traits as recessive. Okay, that's what that question is asking. And then number 11, it says draw an example of a karyotype with trisomy in the space below. Circle trisomy. So when we are drawing the karyotype for trisomy, the only difference between a normal karyotype and trisomy karyotype, which is Down syndrome, is that you will have 3 chromosomes on the 21st set of chromosomes. Okay, so you're going to have 1, 2, 3. Okay, you're gonna do it, set it up like this, and you're gonna have 1 pair, 2, 3, 4, 5, 6 pair. Okay, and we're just gonna put a dotted line in place of the rest of those. But on 21, you will have 3 chromosomes instead of just 2. Okay, and then 22 and 23. So All right. And if you have any questions, send me a comment on Google Classroom. I will try to post this answer key to the study guide on the same assignment, okay, so that you can actually look at the answers yourself, okay? So if you have any questions, just give me a holler.